This is our DNA presentation by Andrew, Anthony, and Tyler.
Oh yeah.
What's up, guys?
The first section is DNA replication. And in general, is the splitting of a strand of DNA into two, semi-conservatively, so that the new two strands contain one old and one new strand. It is an essential process because an old cell needs a strand of DNA, and a new cell that's being split off of the old cell needs a new strand of DNA, and hence why there's two strands that are needed. That's why we need replication, because we need two strands. And all of DNA is made up of the phosphate and nitrogenous base backbone on the diagram that also has all of the, so I guess in reference for this diagram, the yellow and green bars are base pairs that, and in this specific example are the untwisted variants, so it's before strands into the double helix, are bonded by hydrogen, so the base pairs are hydrogen bonds.
Cool. Now let's see. Alright, can you go back to the model real quick? There are a few different things I'd like to talk about here. The first one is the helicase. So in this model right here, it's the blue triangle in the middle. What this enzyme does is it breaks both sides of the DNA bases apart, and it breaks the bonds between the base pairs. Let's see. Now, nucleotide. Nucleotide is a compound consisting of a nucleoside linked to a phosphate backbone.
Okay, and there's several parts to the nucleotide. So it consists of a sugar molecule, which is either a ribose in RNA or deoxyribose in DNA, attached to a phosphate group and a nitrogen-containing base. So these bases used in DNA are adenine, cytosine, guanine, and thymine, A, C, G, and T, respectively. And in RNA, the base uracil, U, takes the place of the thymine. So which enzyme reads each strand and adds to the nucleotides one at a time is the polymerase.
Now, if we go back to the model, if you look, you see the two new strands being created on the left, the lagging strand and then the leading strand. The enzyme, the polymerase, and the DNA ligase add new base pairs to the strand of DNA individually, one at a time. So in this case, the polymerase is the yellow box on the underside of the new two strands being created, and the DNA ligase is the orange box adding up on the top. Now on the lagging strand specifically, there are Okazaki fragments and DNA ligase, which help create new strands for the lagging strand because it's different from the leading strand in that it's the opposite base pairs being added, and so it has to be added in a different manner than the leading strand, and hence why it's the lagging strand is because it's slower. And if you look at the two new strands being created of DNA, you'll be able to see that they're identical to each other, and this is because the old strand is being split into two, and the complementary base pairs are the only kind of base that can be added to a new strand of DNA, and thus, with one half of it, the polymerase adds both of the strands of DNA and both of the base pairs so that it matches them up exactly, and thus creates two identical pieces of DNA.
Yeah, that's pretty cool. It's let's see. It's semi-conservatively because it uses like one strand and it copies that other strand versus like taking sections of the DNA, which would be interesting, but that's not how it works. Yeah. Let's see. Alright, so in the DNA molecule, which is the complementary strand? Let's see. Complementary strands are the ones that are being added. So the new ones that are being added, the little green things right there. So for the top one, it'll be green. The bottom one, it'll be the yellow bases right there. Yeah, Anthony's going to highlight over those. Very nice. Let's see. What other enzymes are involved in this process? Why are they important? So let's see. First of all, we got the primer. the DNA primase, DNA helicase, DNA ligase, and topoisomerase. Sorry, that's hard to pronounce. But yeah, so those are the main ones that we have here. The topoisomerase is the green circle at the beginning. I mean, at the end. What that does is it tells the whole DNA helicase structure to kind of become straight, is to stop twisting and unwind, basically. The helicase, the triangle in the middle, kind of like I already talked about, that one is the one that's actually breaking the base pairs apart. The DNA ligase is the one. The DNA ligase and DNA primase are both pretty similar. The DNA ligase is the one at the top. That one is going to be, pairing them back up, making sure they're all strong and stuff. And the DNA ligase does the same function, but on the leading strand on the bottom over there. So the primer is that little thing Anthony's hovering over, and that one, and it's basically like the prime or the start of it all. That's kind of how it starts. But those are some different functions in the helicase model and DNA replication.
Yeah, and then also DNA is always synthesized in the 5-3 direction as well, and that's labeled here as well on the diagram. So, yeah.
Basically everything is in the 5-3 direction.
Mm-hmm.
Yeah, just like the next topic, which is transcription.
Exactly, transcription. Alright, let's see. So first of all, what is transcription? Well, transcription is the process in which a sequence of DNA is copied into an RNA or mRNA strand that is complementary to the original DNA sequence. Kind of like in DNA replication, where you copy it exactly, only this time you're making an mRNA strand. There are 3 different phases in transcription. The first one is initiation, the second one is elongation, and the third one is termination. So with initiation, the RNA polymerase binds to a part of a gene called the promoter, and then the gene starts to unwind so the RNA polymerase can copy the bases. So if you look in the model, the top part there, that's the first step, initiation. You see the promoter is going to bind to it, and it's going to start to unwind so that it copies the bases. Second step, elongation. The nucleotides are added to the mRNA strand. RNA polymerase reads the DNA copies the base pairs into the mRNA strand. So basically, mRNA is right there. You see the RNA polymerase, and it's copying the strands, and it's not connecting because it's not separating and copying this time. You're just trying to make an mRNA strand. It's kind of like a messenger thing. So it's going to go across it, copy it, and eventually it's going to stop, which is termination, the next step. This step is the end of transcription. The mRNA strand is finished and detaches from the DNA when the RNA polymerase crosses a stop or termination sequence in the gene. That's how it knows when to stop and how it has enough. It's when it hits a stop sequence or termination sequence. When that happens, the mRNA strand is complete. There are many steps inside it, but those are the 3 main steps. So the 3 main types of RNA and the rules respectively. Let's see. So the 3 main types of RNA are messenger RNA, ribosomal RNA, and transfer RNA. They each have their own little acronym: mRNA, RNA, and tRNA. Well, messenger RNA is a transcript copy of a gene that will code for a protein. Ribosomal RNA serves as the catalytic component of ribosomes. Transfer RNA transports amino acids to the ribosome. Makes sense because, it's called transfer RNA.
Yeah. Alright, moving to our other diagram here. So we have the sense strand and the antisense strand. So what makes these different? So the sense strand is a strand of a gene that is not transcribed into the mRNA sequence. So both the polynucleotide strands of the gene travel in opposite directions. The sense travels in the 5-3 direction, which is a common theme, whereas the antisense strand travels in the 3-5, which makes sense because it's the antisense strand. So and you can see that in the diagrams here too. So yeah,
both
the
travel in
antisense
opposite directions. Yeah.
Yeah.
So if you stuck them together, they would make a perfect helix.
Whoa.
Right.
A double helix.
That's why it's called a double helix, kids.
Boom!
So yeah, the antisense strand is the strand of the gene that is transcribed into the mRNA and travels in the 3-5 direction, as we stated, as you can see. So what enzyme binds to the promoter and separates the DNA during transcription. So going back to here, the RNA polymerase is the enzyme that happens to do that.
As stated previously, transcription occurs in the 5' to 3' direction. And to describe the process of post-transcriptional modification, is in essence the process in which mRNA matures. There are 3 steps. Capping. A cap made out of methanol is added to the 5' end of the mRNA strand. This prevents degradation by exonucleases and polyadenylation. The 3' end of the mRNA strand is polyadenylated, which improves the stability and the export of the mRNA strand. And the 3rd step, the coolest step, splicing, process in which introns are removed in order to form a continuous mRNA sequence. A gene is made of introns and exons, and the exons are what code for protein, and since mRNA acts as the blueprint for making proteins, all it needs are the exons. And in essence, removing the introns make the mRNA sequence more efficient.
Exactly, but there's also something else that we should talk about called alternative splicing. And alternative splicing is the selective removal of exons. And I might be thinking, like, what? Because you just said you remove introns, not the exons. But this is just the selective removal of exons, so you're not going to remove every single one. We do this because, well, first of all, it's important because by doing this, a singular gene can create more than one type of protein. Think about it this way. If you had a simple gene, just label it A, B, C, D, easy for example, and first you make a protein using the code ABCD. Then you can remove, say, the C and get ABD, and that creates a separate protein. So you use them all first, and then you can remove some to get more. So in essence, less means more. It's kind of hard to explain in words, but that's kind of what it is. So let's see. Next is gene expression and how it can be regulated. So first of all, gene expression is determined by a level of transcriptional activity. So put simply, higher amounts of mRNA transcripts result in increased amounts of protein. Pretty straightforward. Gene expression can be regulated in multiple ways, but 3 main ways are: 1. Regulatory proteins and transcript factors mediate RNA polymerase action. Nucleosomes regulate transcription, and the 3rd is DNA can be methylated to alter gene expression patterns. But the main way is basically by mediating RNA polymerase action. You can either have one that promotes it or one that decreases the amount of activity, and that's kind of what we're going to talk about next, actually. Transcription factors.
Yeah. So the transcription factors are proteins that bind to DNA to control the gene expression. So this way the body can regulate the amount of transcription that occurs. So activator proteins bind to the enhancer sites and increase rates of transcription, whereas the repressor proteins bind to the silencer sites and decreased rates of transcription.
Basically, if you want to have more, then you add the thing that's going to make it more active. If you want to have less, then you have to regulate it a little bit better, and that's kind of how the body does that.
Yeah. Okay, so moving on to another diagram here. This is the nucleosome and the processes of acetylation and methylation. So the nucleosomes are the finished packages made up of histones and DNA. So the acetylation makes the DNA packaging less tight and more accessible to transcriptional machinery. And then the methylation makes the DNA packaging more tight and less acceptable to transcriptional machinery. So the nucleosomes help to regulate transcription.
A cool little model over here too, shown as a nucleosome.
So here's the histone here, the DNA, and the histone core here.
Yeah, basically the DNA
Nice
wraps
core.
around it, and then the histone, acts as a backbone, which is kind of neat.
Holds it together. Makes
Right.
it a very tight package. What is epigenetics? Epigenetics is the study of changes in organisms as a result of variation in its phenotype, not its genotype, or variations in gene expression levels. And to quote evolution.berkeley.edu, a genotype is the set of genes that an organism carries, whereas phenotype is all of its observable characteristics which are influenced by both its genotype and the environment. such as the sun or radiation or various other environmental factors.
This step is definitely kind of confusing, but I think it makes sense. Yeah.
Yep. The next step is translation. So the process of translation is a process by which mRNA sequences are converted to amino acids. So included with that is something called the ribosomes, which are made of protein and rRNA. And ribosomes are the site of polypeptide synthesis. So it's very important. Here's a diagram to go with that as well.
Yeah, and in the diagram, there are kind of ribosomes and things. So what are ribosomes made up of generally? Well, they're made out of protein and rRNA. Okay, let's see. Transfer RNA in the 4 key regions. So transfer RNA carries amino acids to the ribosome. The 4 key regions include acceptor stem, anticodon, AT arm, and AD arm. This creates a cloverleaf structure, kind of like the diagram on the slides.
Now that we know what tRNA is and have described its 4 key regions, what is it for? It is to help create energy used for peptide bond formation, which then peptide bonds are then linked into a polypeptide chain. But before that happens, tRNA needs to be activated. And the way it's activated is amino acids attached to tRNA via tRNA activating enzymes. Then the enzyme joins ATP to an amino acid to create a charged amino acid AMP complex. Then the acid is linked to a specific tRNA molecule and the AMP is released.
In what direction does translation occur? It occurs in the 5 to 3 direction, kind of like most things that we've gone over. 5 to 3 direction is a common thing. Alright, let's see. Describe initiation. Alright, initiation then. Initiation is the assembly of the ribosomal complex. The steps are 1. Small subunit binds mRNA. 2. tRNA binds the start codon, which is AUG. 3. Large ribosomal subunit binds to tRNA. And 4. Ribosome becomes available to translate.
Alright, and then another step is elongation, which a new amino acid is added to the peptide chain. So the steps of that are the new tRNA enters the A site, and the amino acid transferred from the tRNA in the P site, and then the peptide bond is formed when that comes together. And next would be translocation, which is when the ribosome moves to the next codon position. So the specific steps for that are when the ribosome moves from the codon position in a 5 to 3 direction once again, and then the E site tRNA is released, and then the new tRNA enters the A site as well.
Now, on the next step after translocation and elongation is termination. Ribosomes and polypeptides disassociate from mRNA. The steps are the stop codon is reached, the release factor is received, ribosomes disassemble, and polypeptides are released. Now, the primary structure of a protein describes the order of which amino acids are in a polypeptide sequence. It is formed by a covalent peptide bond between adjacent amino acids, and it controls all subsequent levels of protein structure. The R group allows for grouping by properties, and the amino group NH2 carboxyl group COOH.
Okay, so then the various levels of the protein structure include the primary, secondary, tertiary, and quaternary.
So now there are A helixes and B sheets, but they might seem kind of similar at first. But the main differences are A helix is formed when R group is large, and B sheet is formed when R group is small. So it's basically large versus small.
Pretty straightforward, yeah.
Pretty straightforward, yeah. Alright, another one that are kind of similar, but they're not similar, are the fibrous and globular proteins. The main difference between those is the fibrous protein is long and narrow strands, and the globular protein is compact and round in its shape. So those are kind of different. Fibrous protein, long, narrow, whereas globular proteins are round and compact.
Yeah. And then here's our works cited.
Yeah, we use a lot of notes you took in class for the works cited. We also did a little bit of external research. Those things that we use will be on there. And yeah, besides that, that's the end of our presentation.
Thank you.